Trenton Eddington. So for our experiment, we decided to mess with alcoholic fermentation. Alcoholic fermentation is a biological process that occurs often in nature, and it is simply a process where sugars are converted into chemical energy and CO2 and ethanol are byproducts. So this is the process that is used to make, a lot of consumable alcohol that humans drink. And that is the significance of this experiment. Is what we're trying to do is find whatever pH alcoholic fermentation rates are best at when it comes to yeast. And that is important for, consumers across America, because, alcohol, beer, wine, all those types of alcohol are obviously very popular, and there's a very big market there. So we're just kind of exploring, some of the processes and messing with some of the variables and some of the processes used to make that consumable alcohol for consumers in America and across the world. What we knew coming into this experiment is that yeast ferments sugar into CO2 and C2H6O, which are, CO2 and ethanol. And we also knew that yeast kind of likes a pH of in between 4 and 5. And we found that out from some of our peer-reviewed articles and research that we did before our experiment. pH levels affect the rate of production. That is something that we assumed going into it. And so that kind of helped shape our experimental to design when it comes to, our predictions and research questions and hypotheses and whatnot. But the purpose of the whole thing is to find a good pH that yeast likes, because, going over the labs that we had done and, going through some of the research that we had done, we couldn't necessarily get a clear answer on what pH is the best for alcoholic fermentation rates. So we're basically, adjusting the pH to find whatever pH gives us the best rates of CO2 and alcohol, or of CO2 production, because we're using CO2 data loggers to measure it. So our research question is, how does the pH level affect yeast fermentation rates? Our hypothesis is that highly basic solutions will decrease the rate of production significantly. And our prediction is that if yeast yields lower fermentation rates at extreme pHs lower than 4 and higher than 8, then yeast will produce more CO2 in a solution with more even pH and between 4 and 8. So what we're saying there with our prediction is that as you get further away from 7, which is the perfect balance in between acidity and basic, as you get further away from 7, either towards acidic or basic, the rates will get lower, and the closer that you get towards the center, they will be higher. And coming off, combining that with our hypothesis, we believe that highly basic solutions will have the lowest production rates.
Madeline Evans. The independent variable for our experiment was the pH of the sucrose solutions. The dependent variable was the fermentation rates of those solutions, and the control was the replication with a pH of 7. The materials and methods we used in this experiment, we used pH-adjusted sucrose solutions in glassware, as you can see in the picture, and we made a yeast slurry that we added to those solutions later. We used hydrochloric acid and sodium hydroxide to adjust the pH of the solutions. And to measure the output of our experiment, we used CO2 sensors and data loggers.
Austin Robertson. Our experiment was based on a mixed slurry of yeast. We combined that slurry with the combined pH level solutions of 3, 5, 7, 9, and 11. We mixed these slurries with 25 milliliter slurry and 75 milliliter sucrose. We waited for 3 minutes for a mixture to settle. We tested the CO2 output over a course of 20 minutes. We replicated that 5 times. Based on our findings, we saw that our pH of 3 with 10% yield gave us a 1646.52 output. Our pH of 5 gave us a 2056.474 output. pH of 7 gave us 1638.888 output. Our pH of 9 gave us 2445.974. Our pH 11 gave us 2015.568. Based on these, with our 10% difference, we found no statistically higher rate at any given pH. All our p-values were around 0.95. However, our reps 4 and 5 were skewed slightly by a slightly different temperature of 2 Celsius. This may have played a role in our later experiments.
David McLean. In discussing our findings, we found that yeast's ability to alter pH was a lot more than expected. There's some interesting connotations there. The yeast, across all of our trials, adjusted the pH to approximately 5.5. Everything needed to adjust the pH is there, and it brings the solution into a kind of equilibrium. We expected basic and acidic pHs to retard fermentation. We found the exact opposite. The pH that we started with didn't seem to matter.